Okay, good morning everybody. Welcome to this morning session. We will once again talk about elemental impurities, but I will not bother you with the history of the Q3D. Let's see what is the current situation. Q3D Step 4 was published end of last year, and there are still some training modules to be edited by ICH, normally by the end of this year. Some of them are available as a draft, and if you send me an email, I will be pleased to email them to you. Different organizations are involved now in the implementation of the Q3D. EMA has already stated that Q3D will be applicable June next year for new marketing authorization, while for existing product, it will be end of year 2017. The European Pharmacopoeia will replace the content of Chapter 520, that is currently metal catalyst or metal reagent residues, by a verbatim reproduction of Q3D to be edited in Supplement 9.3 of European Pharmacopoeia. And reference to this Chapter 520 will be made in another chapter, 2619, that is pharmaceutical preparation, that will make Q3D mandatory for all medicinal products. Implementation date is 1st January 2018. USP has already published Chapter 232 and 233 in the second supplement in June this year, and these two chapters will become official in a few weeks. Chapter 231, the heavy metal chapter, will remain in effect till 1st January 2018, but USP already encourages the users to implement the new method as soon as possible. Anyway, 232 will also become mandatory on 1st January 2018 for all USP drug products. FDA, in September this year, has published in the Federal Register a notice of Q3D availability. Once again, implementation date will be the same, June next year for new drug product and 1st January 2018 for existing drug product. And very recently, last week, Japan also announced an implementation date for the new drug product that will be 1st April 2017. It's not clear yet what they will do for existing drug product, but it will come too. So Q3D, the specifications are for drug products only, but the drug product manufacturer will have to carry out a risk assessment. And the information for the risk assessment includes information supplied by drug substance and or excipients manufacturer. In the USP 232, there is a very important sentence for you, API and excipient suppliers. The limits presented in the chapter do not apply to excipient and drug substance. However, elemental impurity levels present in drug substances and excipient must be known, documented, and made available upon request. So, as a consequence, the drug product manufacturers will require from suppliers of API and excipient to provide specification or batch analysis for all their products. Here is the Q3D, 24 elemental impurities divided in 4 categories with PDEs depending upon the route of administration, oral, parenteral, or inhalation. For other routes of administration, you can get information in the training module. They will show you how to proceed. So here is the current situation summary. Once again, implementation for new products, June next year. Implementation for existing product, 1st January 2018. That means that thousands of pharmaceutical products will have to comply with that guideline. Many drug substances and excipients will also have to be analyzed. So In our opinion, it will be impossible to develop and validate one method for each drug product, API, and excipient. So there is a need for a generic method. We have developed such generic method that is applicable to many different kinds of sample. It allows the determination of 23 elements of Q3D, with the exception of osmium. I will come back to that one later. All in a single run. And we have added aluminium to cope with other regulations. We use ICP-MS in order to get very high sensitivity, allowing us to extend the range of the method well below the specification limits. Here are some characteristics of the method. As already said, it's designed for 23 elemental impurities plus aluminium. We use a systematic microwave digestion of the sample with the same mixture of acids. We use high temperature and pressure to ensure a complete digestion of the sample, resulting in the same final sample solution solvent, allowing reduced validation. There is also a control for each sample of the residual carbon content in order to check the digestion efficiency and also to verify that any carbon effect is not present. For the sample preparation, we digest about 200 milligrams of sample. We add internal standard useful for the ICP-MS determination, and we dilute to 40 ml with a suitable solvent. That is a mixture of diluted acids. Calibration of the ICP-MS is done with diluted stock solution that are custom-made for quality assistance with different concentration according to the PDEs of the elemental impurities. The calibration solution are diluted with the same solvent as the sample to ensure a matrix match. That is important in ICP-MS. And all elemental impurities are determined in a single run. I will show in the next slide the range of the method that was designed to cover a very wide range of elemental impurities depending upon the PDEs for each element. The method includes the narrow range recommended by USP 233, namely 50 to 150% of the target limit. But we expanded the range of the method down to much lower concentration. And also for elemental impurities with high PDEs, the range was intentionally limited to 20 microgram per gram to avoid contamination of the ICP. So here is the validated range of the method. Oh, sorry. You can see here the range in microgram of elemental impurity per gram of sample. And in the last three columns, I've presented the maximum concentration of elemental impurities in 10 grams per day for an oral administration, 10 grams per day for a parenteral, and 1 gram per day for an inhalation product. And you can see that these maximum levels are well within the range for each elemental impurities. The method was fully validated for 3 different compounds: the BSA, representing the protein world, cellulose, representing the polymers, and carbamazepine, that is a very difficult drug substance to digest. The acceptance criteria are based on USP 223, European Pharmacopoeia, and also some of our internal procedure. After the complete validation of the method, we verified the performance of it for several other matrices. Some product difficult to digest, polyethylene glycol, tannic acid, phenylalanine. Some organic acid, tannic and citric acid. 2 organic bases, caffeine, triethylamine. Some inorganic salts, sodium chloride, sodium carbonate, potassium phosphate. A compound able to complex metal, cysteine. A metal-containing excipient, magnesium stearate. and a finished drug product, paracetamol tablets, sorry. So here are the validation results. We demonstrated that the specificity is okay versus digestion mixture and dilution solvent, but also versus the sample matrix for the BSA, the cellulose, and carbamazepine. We demonstrated that the recoveries for spiked sample meet the USP precision and accuracy acceptance criteria. But we also verified the specificity of the method versus major elements that are elements that are not in the Q3D, but likely to be present in high concentration in some pharmaceutical samples, like aluminum, calcium, iron, and so on. And we have shown that in presence of very high concentration, 2,500 microgram per ml of these major elements, the recoveries of the Q3D elements are still within the acceptance criteria. Linearity of the method was very good for all elements. And we evaluated the accuracy and precision for BSA, cellulose, and carbamazepine with sample spikes at 6 different levels, covering the whole range of the method, with 3 sample prep at each level and even 6 at mid-range. I will show you that all recoveries are well within the acceptance criteria, 70 to 150%. The RSDs are less than 30% at the lower limit of quantitation, while it is lower than 20% for all other levels. Intermediate precision was also assessed using the same instrument on a different day with a new independent calibration and new sample preparation of spike and unspiked. Once again, RSD was less than 20%. The solution is at least 24 hours, allowing re-injection in case of problem. In this table, you can see the accuracy, precision, and intermediate precision for BSA. Result obtained on day 1, day 2, and combined for day 1 and day 2. You can see that recoveries are very close to 100% on each day, and the RSDs are quite acceptable. The main figure are here. Minimum recovery is 92% and maximum is 102, with an RSD that is always less than 5%. Same results for cellulose. Once again, recoveries are ranging from 95 to 106%, and RSDs are still less than 5%. And finally, for carbamazepine, once again, very good recoveries, and RSDs lower than 5%. Now, we verified also the performance of the method with 12 different matrices. For each sample, we prepared 3 unspiked and 3 spiked samples, and we calculated, we measured the recoveries and RSDs of all elements. The result will be presented in the next slide. And we also checked that the residual carbon content was always less than 0.1 milligram per ml. Here are the results of the performance verification for polyethylene glycol, phenylalanine, tannic acid, and so on. You have always the recoveries and the RSDs. We will not review all the results, but once again, always close to 100% with very good RSDs, lower than 5% for most of the elemental impurities. And here are the results for 6 additional matrices, the inorganic salts, triethylamide paracetamol tablets, and magnesium stearate. And once again, you can check that the recoveries are well within the acceptance criteria with very good RSDs. So we have validated the generic method completely for 3 different compounds, BSA, cellulose, and carbamazepine. The performance of this method was also verified for 12 very widely different matrices. Remember that systematic digestion will bring all samples in the same final solvent, allowing reduced validation work. In fact, we propose a simple verification of performance rather than a complete validation for a new sample. So this method was extensively validated and guarantees reliable results and offers a sufficient degree of confidence for finalization of your risk assessment. Here is our proposition. For an API or an excipient, using the generic method, we suggest to analyze 3 batches of the API or excipient taken at the end of the shelf life. In such way, all potential sources of elemental impurities will be taken into account, even coming from the manufacturing process or from leaching from the container closure system. For minor excipient, you may require more than 3 batches to analyze to take into account the high variability inherent to that type of natural product. And in some cases, analysis of all batches may be required. For an existing drug product, once again, we suggest to analyze 3 batches taken at the end of a shelf life for the same reasons. If you can demonstrate that all elemental impurities are below 30% of the PDE, then you are lucky. You are done. You don't need any additional control. You just have to document. That's all. If one or more elemental impurities is between 30% and 100% of the PDE, then try to identify the source and reduce the elemental impurities. Or you will have to establish specifications and controls. In that case, a specific validated method will be required. And if you are unlucky, you have 1 or more elemental impurities that is above the PDE, then you have to reduce it or try to justify higher level. In some cases, it is allowed by Q3D. However, you may have to change the quality or the supplier of the component that is the source of the problem. You may have to change manufacturing equipment that might be responsible of the contamination, or you may have to change the container closure system. For a new drug product, try to select the component and the container closure system on basis of the supplier information. If such information is not available, once again, you may have to carry out a screening of elemental impurities, and our method is also suitable for that purpose. Remember that early assessment of elemental impurity in the formulation will avoid non-compliant drug product. And what about elemental impurities from the manufacturing process? Then, once again, the only way to be sure is to analyze some batches of the drug product. And for the elemental impurities potentially leached from the container closure, it will be addressed during the stability studies. Some words about osmium. Osmium is the least abundant element in the Earth's crust. So it is very unlikely that you will have contamination in your pharmaceutical product. However, if osmium has been used as a catalyst somewhere during the synthesis, then you will need to determine the residues of osmium in your product. And this is a challenge. Why? There are some reasons to underestimate the osmium. Usually, it is used as a catalyst in the form of OSO4. That is a volatile compound that is also formed when you digest your sample with nitric acid. OSO4 sublimes at room temperature. The boiling point is very low, 130 degrees. So digestion of sample in an open vessel will result in near zero recoveries. It is also a very non-polar product. It loves plastic like PTFE or PFA, in which it penetrates very easily. So microwave digestion in closed vessel made of such inert polymers will also result in low recoveries. And additionally, you will have large memory effect in the next sample. You have also reasons for overestimation of the osmium by ICP. When measuring the OSO4, during a nebulization, it volatilizes and it goes into the gaseous phase and is taken to the torch by the carrier gas, resulting in an excessive amount of osmium going into the ICP. And you may have recoveries, apparent recoveries, that go up to 600%. And to complete the picture, OSO4 will also stick to the plastic tubing, generating high memory effect for the following samples. So it's really a challenge to measure osmium. Knowing all these drawbacks, we have developed a method that is suitable for a wide range of samples. We use, once again, systematic digestion of sample, but in high-pressure glass vessel. Before digestion, we spike sample with 190 isotope of osmium to allow the calculation of total osmium content by reverse isotope dilution techniques. And that will compensate our losses or under or overestimation. The range of the method is from 0.2 to 2 microgram of osmium per gram of sample. In final conclusion, we have validated method for the determination of all Q3D elemental impurities that are applicable for APIs and excipients in the validated ranges, for oral drug product with up to 10 gram daily dose, for small volume parenteral products with up to 10 gram daily dose, and for inhalation drug product with up to 1 gram daily dose. Obviously, custom-made method may also be developed and validated for other products, like large volume parenterals, up to 2 liters per day, for example. We have a lot of experience with such products too. Finally, this methodology is now well implemented in quality assistance. We have 5 ICP-MS and 2 microwave digestion systems that allows fast and cost-attractive analysis of your sample. So here's the question time. If you want, you may visit us on our booth, 3H70, or you can send me an email. Once again, if you want the training module of the ICH, I have already several of them available. And you may even have a look to the presentation within a few days on our website. Now, if you have any question, I will be pleased to answer.